it is highly technical, but at the same time, this is their proclaimed best and most technical line of evidence for universal descent. So I believe it deserves the attention. And we talked a lot about protein moonlighting earlier. We went over just some of the literature, even though there really is massive amounts of literature that gives us at times unexpected functions of RNA. The fact that we functionally tested based on genetic knockout tests very, very little of the genetic language. We understand very little of the genetic language. Jeremy, you and I always talk about that. I like giving the analogy. It's kind of like if I were to go to Russia, and Jeremy, you used this before too, and I can't speak any Russian, and I went over to Russia to be a proofreader, and I looked at a book of 200 pages and I had to proofread it. I'm not going to know what's a mistake. I'm not going to know, just like when we're looking at the genetics, the pseudogenes, for example, they'll say is, these are shared genetic mistakes. But that's analogous to me trying to point out as a proofreader in Russia, even though I can't speak any Russian, what is a mistake and what is not. That's how little we know of the DNA language. And the fact that we predict that the vast majority of these DNA differences are functional, and evolutionists assume that they're all the result of mutations over time, that notion that the evolutionists have is slowly being overturned. Classic paradigms are being overturned. And that's why Herman Mayes, and just to represent their position fairly, Mayes will often look to, like, he's had numerous debates with Hovind, Jensen, and Jackson, and he used the same argument in every single debate. He'll look to a large number of gene trees and he'll say that they all corroborate with each other. He'll point out that in phylogenetics, the best thing to look at is neutral variation. For the audience sake, that's just variation in DNA.
I don't agree.
Pardon, Jeremy?
I don't agree that we should look at neutral variations. We should look at the numbers that I presented.
No, I know. I'm just saying that this is what he says. I'm representing what he said.
He's trying to fool everybody.
And I rewatched his debate with Hovind earlier, Kent Hovind, so I know this is exactly what he said. And I rewatched his with Jensen and Jackson recently too. So I'm aware of his arguments. But you're right. Yeah, he'll say that that's the best. Conspiracy Cats, who I debated too, he'll say that the hierarchies in the cytochrome C protein, that's the best. So they've all got their own best, but it doesn't matter because the bigger picture is nested hierarchies, right? Herman Mayes is looking at neutral variation in regards to third position, codon variation. Conspiracy Cats is looking at mitochondrial DNA proteins. Regardless, it's all hierarchies is what they're looking to. So Mayes specifically, he's looking to that type of variation in our DNA, and this applies universally across species, that doesn't vary with function. Now, if the audience has been listening, right off the bat, we know that the evolutionist assumes lots of variation in DNA is neutral and non-functional to begin with. We don't make that assumption. So Mayes, his type of neutral variation, as I said, is the third position codon variation, for example. So to make it simple, we go from DNA to RNA to a polypeptide, and each amino acid, okay, it's encoded by a set of 3 bases. That's what we call a codon. We know that there are 20 amino acids that are encoded by 64 codons. Now, I Googled this just to make sure, just so I'm not guessing. I Googled valine, okay? We could have up to 4, GUG, GUA, GUC, and GUU will all make valine. All 4 of these codons will make valine. This means the third position can change and yet have no effect on phenotype because the amino acid it's actually encoding is exactly the same. That's his point, okay? So these types, among others, is what somebody like Herman Mayes would say are the best types of variation to look at when producing phylogenetic trees, because he's saying if there was a common ancestor, then these neutral changes must be passed on from those ancestors. Selection's not acting upon these neutral variations because neutral means it's neutral to selection. Okay, so he'll point out that this observation on neutral variation cannot be true if independent ancestry is true. But the fact is, far from being neutral junk or just evolutionary leftovers over deep time, these so-called, degenerate synonymous codons actually and amazingly represent an independent secondary code in DNA. So even this paradigm is being overturned because they're saying that there's a lot of redundancy in DNA coding for protein. We went over the fact that redundancy is good. It's good in computer code. It's good in it's good in the DNA code. But now we know that although statistically, because overall this is statistical hierarchies is what Mays looks to. It can be variable, but we're finding, and I've got papers here in front of me that show this, we're finding that it actually plays an additional functional role. They're not just redundant elements. They're not just tolerating error. The cell is actually using different redundant elements to slow and speed the process. It's information compression into every single letter, and it's it really is fascinating. So although these multiple codons, as I went over, may code for the same amino acid, the codons themselves have other functions. And the specific codon you're looking at is important, oftentimes specific to the kind of creature you're looking at. So once again, for the audience, boom, we both expect nested hierarchies. That is the rule. But what can differentiate between those hierarchies? Well, as Jeremy pointed out, we can look at the single nucleotide differences oftentimes, and we can look for gaps, which we've done. We can do that with the CO1 gene. And function. Function's key. But most importantly, what model is making the best testable predictions? What hypotheses has the best testable predictions that flow from them? And it is the creationists that are making the testable predictions on third position codon function. Dr. Jensen has like a 30, 40 page paper outlining very specific functional predictions on cytochrome C mitochondrial DNA differences, third position codon functional differences as well. So genetics is where we win. And I believe at this point, genetics is where we are winning. So I'm pretty happy to say I think we've addressed every single one of their best lines of evidence for hierarchies. If you do have one last thing to say, Jeremy, to add to that, to be